Hello everyone. Let us start our today's science class with the thought: let your smile change the world, but don't let the world change your smile. We have started lesson number 10 and we are studying various cell organelles. The next cell organelle after mitochondria is vacuoles. Vacuoles are the storage sacs for solid or liquid contents. They do not have any typical shape or size. The structure of vacuole changes according to the need of the cell. Vacuole is bound by single membrane. Vacuole acts as a storage sac and it contains solid or liquid material. Vacuoles change their shape and size as per the need of the cell. Thus, they do not have any fixed shape or size. And vacuoles are bound by a single membrane. Functions of vacuoles. Number 1, to maintain the osmotic pressure of the cell. Children, do you remember the definition of osmosis? The traveling of water from a part with more water to a part with less water through a selectively permeable membrane is called osmosis. It is a physical process and it do not it does not consume cellular energy. The best examples of osmosis process: number 1, uh, you can try this at home. If you keep the resins in water at night and if you observe it in morning, you will see that they all are swollen up. What had happened here? The water content inside the resin is less as compared to water content in the outside medium. Here water enters in the resin due to the process of endoosmosis. Clear? On the other hand, if resins are kept in sugar solution, they show plasmolysis and they shrink. The sugar solution acts as a hypertonic medium. Water content in the resin is higher than that is present in sugary solution. Thus, water exists from resins and its content thus shrinks. Similarly, one more example you can easily observe in rainy season. During rainy season, there is more humidity in air. The doors get soaked in rainwater. Though wood is non-living, it has ability to absorb water. As the moisture is more in the surrounding area, it enters the wood. This is a type of endoosmosis. It causes the doors to swell. The swollen doors then fit very tightly. Children, I am actually trying to explain what is the osmosis process and what are the are two different types of osmosis: exoosmosis and endoosmosis. Once the concept of osmosis is very clear to you, you can understand the function um, of vacuoles. So vacuoles are maintaining the osmotic pressure of the cell. Clear? Once the cell become uh, swollen up, its cell wall becomes turgid, isn't it? So vacuole is actually helping to maintain the osmotic pressure of the cell. Number 2, to store metabolic byproducts and end products: glycogen, proteins, water. During the process of photosynthesis, carbohydrate is formed. So this is a product, means metabolic product which is formed due to the process of photosynthesis. So these byproducts are stored in vacuoles. In animal cell, they store waste products and food, while in amoeba, it stores food before digestion. In plant cell, vacuoles are full of cell sap and provide turgidity, rigidity to them. Now, we will see next cell organelle, plastids. The organelle which gives colors which are seen in plant parts. Have you seen green leaves, yellow, red, orange, blue flowers? Huh. The organelle which gives such colors is present only in plant cell and it is called plastid. Plastids have double membrane and they are of two types: leucoplasts, white or colorless plastids, and chromoplast, colored plastids. Chloroplast can get converted into the other type of chromoplast. For example, raw green tomatoes turn red due to breakdown of chlorophyll and synthesis of lycopene. Plastid is a cell organelle which is present only in plant cell. It is present only in plant cell. It has double membrane, outer membrane and inner membrane. You can easily see here outer membrane and inner membrane. Then thylakoids present which as grana and stroma, in which photosynthetic pigments are present. Grana is a plural of granum. Thylakoids are the disc-like structures in which photosynthetic pigments are present. Stroma contains enzymes, DNA, ribosomes and carbohydrate that are necessary for photosynthesis. This is the structure of chloroplast. There are three types of plastids: chloroplast, green plastids. Chlorophyll converts solar energy to chemical energy during photosynthesis. You all know the process of photosynthesis in which solar energy from sun is converted into chemical energy. And who is going to help in this process? Chlorophyll. How red or how raw green tomatoes they turn into red color? When green tomatoes become ripe, they lose chlorophyll and develop red pigments in them called lycopene, and therefore ripe tomatoes appear red. Clear? Green color of the plant part is due to chlorophyll. Orange color in carrot is due to carotene. Yellow color in flowers is due to pigment xanthophyll. Purple blue color is due to pigment anthocyanin. And dark pink, beetroot has dark pink color and it is due to betal betal bet, betalains. And one more uh, type of plastid is there, that is called as leucoplast. Leucoplasts are involved in the synthesis and storage of food like starch, oils and proteins. So three different types of plastids are there: chloroplast, chromoplast and leucoplast. Chloroplast converts solar energy to chemical energy. Chromoplast give different colors to flowers and fruits. Leucoplasts are involved in the synthesis and storage of food like starch, oils and proteins. Is it clear to you all? We have discussed the structure of chloroplast. Here It is an activity given. Take out a thin peel of Rio or croton leaf and observe the chromoplast under the compound microscope. Chloroplasts are important for the photosynthesis process that takes place in the leaves. Chlorophyll in chloroplast traps solar energy and converts it to chemical energy. Stroma in the chloroplast contains enzymes, DNA, ribosomes, and carbohydrate that are all necessary for photosynthesis. Mitochondria and plastids can produce proteins as well as replicate themselves as they have their own DNA and ribosomes. National Center for Cell Science (NCCS) is an independent institute working under Biotechnology Department of Government of India. It is located in the campus of Savitribai Phule Pune University Pune and involved in research in cytology study of cell. It provides services for National Animal Cell Repository. It is mainly working on research about cancer treatment. National Center for Cell Science (NCCS), which is working under the Biotechnology Department of Government of India. It is located in campus of Savitribai Phule Pune University Pune and it is involved in the research of cytology. We will see the difference between eukaryotic cell and prokaryotic cell in next video.